right, let's see. Is the audio working? Test. Yep. All right. Hey, how are you guys doing? Let's go ahead and jump in. So, the last time we talked, we started by talking about coronavirus. And let's see, it's got a new name. Have you seen? It's COVID-19. That's an official name. Coronavirus was really the type of virus that it was. It wasn't the name of the specific virus. And so COVID-19, I gave you a bad number when we talked about it. I said 8,000 people died last year. It's actually 8,000 people so far this year have died from the flu. In a bad year, the flu can kill as many as 60,000 people in the U.S. So those are some pretty striking numbers. New research from epidemiologists this week shows that COVID-19 is actually far more dangerous than the flu. If Let me switch views here and pull up a calculator. All right. So the death rate for the flu is about 1 in 1,000 or 0.1%. Okay, so if we convert that into a decimal, that's 0.001. You know, there are 300. Back up a second. Multiply that by the population of the U.S., which is 330 million. If everybody in the U.S. got the flu, now not everybody in the U.S. gets the flu, but if everyone in the U.S. gets the flu, 330,000 people, statistically speaking, should die from it. Um, on the other hand, COVID-19 has a death rate of not 1 in 1,000, but 20 in 1,000. That's also 2%. So if we convert 2% to a decimal and multiply that by the population of the United States, Uh, if everyone in the U.S. got COVID-19, that would result in a death toll of about 6.6 million. All right, those are some pretty striking numbers. So, yeah, we should definitely get this under wraps. It is slowly making its way into the U.S. As you know, there have been a couple confirmed cases. And, we don't all necessarily need to run out and buy flu masks today, but just do be conscientious. When other people are sneezing and coughing near you, maybe cover your mouth. And of course, when you cough or sneeze, cover your mouth to prevent the spread. All right, because you never know who might be infected. And again, you may not even know if you're infected, may feel like the flu. Symptoms are pretty similar. Um, all right. So that's enough doom and gloom. Let's talk about mitosis and meiosis. So to start, let's take a look at DNA because mitosis and meiosis, they're an important step in dividing the cell, but the key to mitosis and meiosis is how we divide DNA. So let's look at DNA. We covered the macromolecules and their subunits a while ago. But if you look at this pattern over on the right, okay, you've got this familiar shape. You've seen it before. Science-y movies use it all the time. It's very prevalent in the Resident Evil movies. This is known as the double helix. A helix is a corkscrew pattern, and the double helix is this pattern with the two corkscrews wrapped around each other, okay? And so this is how DNA looks really, really, really up close, okay? Each of these colored bars is a nucleotide. This is how it looks way, way, way zoomed in. In fact, our electron microscopes, I don't think are powerful enough yet to show us this exactly. But anyway, so if you were to zoom in, now you zoom out a little bit, and it starts looking like this string. Okay, it starts looking like this string. And you see these green balls. Those are called histones. So histones are a protein that we wrap the DNA around. Why would we do that? Well, to conserve space. If we had DNA sitting out loose, or we might say relaxed, okay, just without the histones, it would take up a lot of space. We wrap it around the histones, and that helps condense the DNA so that it takes up less space. Also, When DNA is wrapped around a histone, any genes that are in that region that's wrapped around a histone can't be accessed. So there are protein machines that come along and they copy genes, they copy sections of DNA into mRNA, and then those messenger RNAs get sent off and they get translated into proteins. But if that gene, if that section of DNA is wrapped around a histone, then it can't be accessed by the transcription machinery, by those protein machines that copy it. And so this is actually also a way for us to turn turn genes on and off. We can turn a gene off by wrapping it tightly around histones, and we can turn it on by unwrapping it and releasing it. Kind of an interesting thing. You probably know, you may know that the thing that makes human females, female, is that they have two X chromosomes, whereas the male has an X and a Y chromosome. And if you have too many copies of a gene, you may run into gene dosing issues where you just are making way too much of certain proteins, and that can actually be bad for the cell. And so the way females deal with that is to wrap one of their X chromosomes up very tightly. We would say it's highly condensed. All right, wrap it up very tightly around these histones so that transcription machinery can't get to the DNA. Now, again, females have two X chromosomes per cell, and we would just wrap up one of them permanently. Now, which one gets wrapped up is actually random. So from cell to cell, it may be a different X chromosome that's active. Interesting thing about that is, let me go ahead and pull it up. At least an interesting example of it are calico cats. Have you ever seen one? Calico cats are only female. Okay, they're only female. That's pretty cute right there. And so what happens with the calico cats is that the gene for fur color, either black or orange, is on the X chromosome. And so certain regions of the cat will have one X chromosome active. Say in this region, it'll have the orange containing X chromosome active, and the black one will be inactivated. Over here, where the fur is black, you can see that the X chromosome with the black gene is it's exposed, and the orange X chromosome is condensed. It's wrapped up around histones, and so you can't access the orange gene in this region. All right, so that's a interesting example of that, I guess. Let's go back to the images. All right. So it's normal though for us to sometimes wrap our DNA up like this. We're going to wrap our DNA up like this where it's tightly condensed and you can't get to any of the genes. We're going to condense it until it looks very tight, like this large structure that we call a chromosome. Okay. Now, we do this when we're packaging DNA up and getting ready to divide it during mitosis or meiosis. All right, so we wrap it up tightly, we package it up temporarily. This is for all other chromosomes, temporarily, so that we can easily divide it and then split it between the two daughter cells. All right. No questions so far. Okay, let's keep moving. Okay. Let's talk about the cell cycle then. Zoom in a little bit. So here's a diagram of the cell cycle, and from the size of each quadrant, it's divided into 4 large sections. From the size of them, you might guess that they take roughly the same amount of time. That's actually not true. A cell will spend the vast majority of its life in interphase. Okay. Now, inter means between, so this is the phase between, growth cycles and between uh, mitotic divisions. Okay, so let's go over here and start at the beginning. We've got phase G1, or the first growth phase. Okay, and this is where we're going to see growth of the cell. We're going to build the cell, make it larger. Okay, and then once the cell is grown and reached its normal full size, okay, we will enter interphase. And in interphase, we're going to do all the normal activities the cell would normally do, and it's going to spend most of its life in this phase. When it's ready for division, when it receives signals to divide, it'll enter G2, or the second growth phase. And in the second growth phase, we build up our organelles, we take on more cytoplasm, take on more fluid, so that we can get ready to divide the cell. Finally, we move on to this region, and I want to point out something here. A lot of people talk about mitosis as if it were cellular division, and it's actually not. Mitosis is followed by cytokinesis. Cytokinesis is actually cell division, and it takes place immediately after mitosis. Okay, so a lot of people are going to talk about mitosis like it means cell division. It doesn't. Technically, mitosis refers to division of the nucleus. We are dividing the nucleus. We're going to form two new nuclei, and then immediately after that, we divide the cell through cytokinesis. Now, there are 4 stages to mitosis, and they are prophase, metaphase, anaphase, and telophase. And as you can see, those begin with the letters, and they're in order. They begin with the letters P, M, A, T. And the acronym I've always used, or the mnemonic device, I should say, that I've always used to remember that, it's very stupid, but PMAT, just PMAT. You know, like the kind of mat you might set out for a puppy who hasn't learned to go outside. It has nothing to do with the functions of mitosis, but it's always helped me remember the order, PMAT. Okay. All right. So we've talked about this. Now let's talk about which cells, I guess, do go through mitosis. All right. And so, the cells of your body can be largely divided into 2 main types. So you've got somatic cells, and these are the adult cells. Most of the cells of your body are going to be somatic cells. Some of them will go through mitosis, and some will not. All right, and so skin cells, your outer layers of the skin, are made up of what's called epithelium. Your mucous membranes are lined with a type of epithelium also. So epithelial cells, they will divide your entire life. What happens when you get cut? Your epithelial cells are going to divide and replace the old ones that are missing, and then they're going to fill in the gap and close the wound. All right. You probably know if you don't shower or scrub your skin, you may see in some regions, particularly around the face, an accumulation of dead skin. All right, so what you should know about that is that the bottom layer of the skin is constantly dividing and producing new layers of skin. And each time the bottom layer divides, it pushes the next newest layer up and then up and then up, all right, toward the outside of the body. That's an important feature of the skin, and also your mucous membranes in the mouth and like in the anus and vagina as well, is that they can come into contact with things and they can sustain friction without damaging the tissue. Basically, when a solid object comes into contact with any of these tissues, let's say the skin, for instance, I bump into a wall, I can slough off or lose the top layer of cells, and that's okay. I'm protected because I can lose that top layer of cells. Being able to lose that top layer of cells is also important because sometimes, actually all the time, we have bacteria growing on our skin. And as I lose cells from the top layer of skin, as they fall off, they take bacteria with them. Now, not all skin bacteria is bad, but some is. All right, good. So those tissues will constantly be dividing. We have other tissues, like connective tissues, which can divide, but they do so usually pretty slowly, depending on which type of connective tissue. Um, Red blood cells cannot divide because they have no nucleus. They're the only human blood cell that has no nucleus. However, the cells that make red blood cells do have a nucleus, and they divide constantly to continue producing red blood cells. Okay, now there are a couple other cell types that do not divide. And so you've got neurons and muscle cells. So when you go to the gym and you're working out, okay, and you do this, it's a new habit, new year, new you, you keep working out, and your muscles increase in size. And it's like, golly gee, how'd that happen? Well, you're not producing new muscle cells. What's happened is that the existing muscle cells have enlarged. Okay, they've gotten bigger, they've gotten more efficient, but you're not developing new muscle cells. And so there's a process called atrophy, and we could say, let's talk about disuse atrophy. That's where, for whatever reason, you lay in a bed for years at a time, and then over time, your muscle cells are replaced with connective tissue. Well, once that's happened, you're not going to grow new muscle cells, and so the damage is irreversible. This is particularly evident in another type of atrophy called denervation atrophy. If you're stuck in a bed, you have some movement. Some movement is possible. But if you have denervation atrophy, which is where the nerve that stimulates a muscle has been damaged or destroyed, that muscle will never contract and it will wither away. It will be replaced with connective tissue again. And even if you somehow could repair the connection between the muscle and the nervous system, or somehow stimulate that muscle with electricity, it would not contract because the muscle cells have been replaced with connective tissue, and there's no treatment for that because you can't regrow muscle tissue. Same with the nervous system. Some nerve fibers, under certain circumstances, if they've only been partially destroyed, can regrow themselves. This one muscle, or this one, nerve fiber can, under certain circumstances, rebuild itself. However, you are not going to build new neurons to replace a damaged nerve fiber. You can't grow new ones. All right, so those tissue types do not divide. The ones that do, mostly epithelium, some connective tissues, they'll do it. All right, what else do we have? Oh, the other type of, the other cell type, those were all somatic cells. Some of them divide, some don't. The other type are the sex cells, or germline cells. And we'll talk about those in meiosis in just a little bit. They're a little bit different. They have half the number of chromosomes as somatic cells, and they go through a separate process, meiosis. All right, so we've talked about the cell cycle. We've introduced mitosis. Let's go ahead and, okay, go ahead and jump into mitosis. Okay. Something strange is happening on my other screen. Uh-oh. I just made everything disappear. Okay. Good. Good. All right, I can see it again. All right, so here we go. There we go. Let's zoom in so you can see this better. All right, so these are the stages of mitosis. Over here on the left, they show you interphase. It starts with interphase, but actually it starts with G1, then you move into interphase, then you move into G2, then you can start mitosis when you receive the signal to divide. All right, so let's go ahead and start mitosis over here. It's labeled 2, but that's kind of out of order. Prophase is the first step, and there's an early prophase and there's a late prophase. In early prophase, you're going to see the DNA start winding up, start condensing, and it's going to look like a bowl of spaghetti here, all right? And so you're seeing it start to condense, you're seeing it start to wind up, to wrap up, to condense. In late prophase, you're going to see that the DNA is condensed and it's distributed throughout the cell. Sorry, throughout the nucleus. You're also going to see, you see this dotted line out here, you're going to see the nucleus begin to dissolve and actually disappear in prophase. And the next step is prometaphase, where the nucleus has broken apart, you can see. Okay. Then we move from here into metaphase, which is for some reason labeled metahaze right here. And anyway, in metaphase, that's where the chromosomes are going to line up down the middle. And so if you see a picture where they are lined up down the middle, that is metaphase, the second step. All right. So you can see here also the mitotic spindle forming. Okay. You've got the centrioles, which are these giant protein anchors made out of microtubules. Okay, those are the centrioles. And connected to them are cytoskeletal elements that are going to move the chromosomes around. Okay, so we move the chromosomes around in metaphase, we line them up down the middle. Anaphase is where we're going to pull and separate the chromosomes. Okay, and then telophase, telo means last, so this is the last phase. This is where we're going to reform the nuclei, okay? Begin to reform the nuclei. And then you can see this cleavage furrow here between what will become two daughter cells. Here's cell 1, here's cell 2. So immediately after mitosis is done, these two cells are going to pinch off and separate, and then that will be the end of cytokinesis. Okay, cytokinesis is the actual division of the two cells, and so cytokinesis will follow mitosis by pinching off this little cleavage furrow right here. All right, was there anything we forgot to mention? Let's go back and talk about the chromosomes for a second because I want to point something out here. All right, so we're wrapping up DNA. Okay, we're wrapping up DNA, and if we go back to the cell cycle, during the second growth phase, we're going to be replicating the DNA. No, that's not true, sorry. We're going to do that during the synthesis phase or S phase of interphase. We're going to replicate the DNA, that's right. We're going to be replicating, sorry. We're going to be replicating organelles during G2. Anyway, we replicate the DNA, we double the amount of DNA we have, and then we go back here, we wrap it up, and we're going to get this structure called a chromosome. Now the chromosome is actually made up of two sticks. We could say this is the stick on the right, and here's the stick on the left. Each one of them is called a chromatid. Remember, chromatin is DNA wrapped around histones. A chromatid is this highly condensed form of the chromosome. So we have two chromatids. Here's one, and then here's two. There's one on the left, one on the right. And they're joined together at this anchoring point called a centromere. All right, the centromere anchors two sister chromatids here together, okay? And so, good. In metaphase, we're going to line up the sister chromatids down the middle. Okay, and then in anaphase, we're going to pull them apart. Okay, we're going to pull them apart. Let's pull up another picture just to kind of make our point clear here. All right, let's do karyotype. Here's a cool little picture. All right, so humans have 23 pairs of homologous chromosomes. Okay? Homologous means that they correspond to one another. So this is a chromosome 1, this is a chromosome 1, this is a pair of chromosome 1s. Over here, this is chromosome 2, this is a different chromosome 2, and they're a pair of chromosomes that correspond to each other, so we call them homologous chromosomes. All right, so we have 23 of these pairs. Okay, you may have heard of one of those DNA sequencing companies. They're called 23andMe. It's one way to remember we have 23 pairs of chromosomes. All right, and so they're numbered by size. Chromosome 1 is the largest. Chromosome 2 is the next largest. Chromosome 3 is the next largest, and so on down to chromosome 22. The 23rd pair of chromosomes are the sex chromosomes. And so in this example, they have two X chromosomes. All right, two X chromosomes. See, this should be, yeah, right here's an X and a Y in this other image. Here's an X and a Y. As you can see, the Y is smaller. The Y doesn't have a lot of information on it. However, it does have the genes that encode maleness. So in the absence of the Y chromosome, say you had two X chromosomes or you had just an X chromosome, you would be female. You are only male, biologically speaking, in the presence of the Y chromosome, because the genes that determine maleness, the development of the testes, the external genitalia, and so on, those genes are all on the Y chromosome. It's small, doesn't have a lot of information. The little amount of information it does have is mostly dedicated to determining male features. So let's go back to this picture because it's prettier. All right, so you can see they're all lined up here. They get smaller all the way down to 22. One interesting thing about the human chromosomes, we have 23 pairs. Our closest living relatives, the great apes, have 24 pairs of chromosomes. Now, why is that? Well, it turns out that if we use human numbers to describe ape chromosomes, humans have a chromosome 1, and then we have chromosome 2. Apes have chromosome 2A and 2B, and our human chromosome 2 actually resulted from the fusion of ape chromosome 2A and ape chromosome 2B. Pretty wild stuff. So this is possibly part of what drove human divergence from our ape relatives in the recent past. In addition to the FOXP2 gene responsible for speech, which is on chromosome 7. That last bit was just interesting trivia. Anyway, good. So we've talked about the chromosomes. And so if this is called a karyotype. This image is called your chromosomes, and then using imaging software, they line them up to check for any chromosomal abnormalities. Down syndrome you may have heard of. That's a gene dosing disease. And in Down syndrome, which is also known as trisomy 21, the individual has 3 copies of chromosome 21. That's why it's called trisomy 21, means you've got 3 copies of chromosome 21. And I'm not sure which proteins exactly are responsible, but some of the proteins that are on chromosome 21, people with Down syndrome are getting too many of those proteins. And so that leads to the developmental changes that we see in people with Down syndrome. I think I called it a disease. Some people would probably take issue with that. We can say condition. It's different. Yes, developmentally different. Not to place any judgment. All right, so let's go back to the picture. Picture. All right. So here we have metaphase again. Actually, we'll talk about trisomy 21. We'll talk about that. It's not a mitotic condition. It's actually a meiotic condition. So let's move on then and get to meiosis. So meiosis is the process of producing dividing the nucleus in the production of sex cells. Okay. So male sex cells, of course, we know they're called sperm. Female sex cells are called eggs or oocytes, right? And so we mentioned we have 23 chromosomes. That is, you could say we have 23 numbered chromosomes, 23 chromosomes. This chromosome 1 does not contain the exact same information as this chromosome 1. They all have the same genes, okay? All of the genes that are supposed to be on this chromosome, they're going to be on the other chromosome too. But you can have different versions of genes. And so, for instance, people have different hair colors, And so one version of, let's just go ahead and simplify it and say one version of the hair color gene might be on this chromosome. Let's say it's the blonde version. And the version for black hair may be on this chromosome 1. In both cases, the gene for hair color is on chromosome 1, but you can have different versions on these two. We'll talk more about that in a little bit. Now, I should just give a disclaimer. I made up that example off the top of my head. Don't know for sure that hair color genes are on chromosome 1, so don't quote me on that. All right, so moving on. We have 23 specific chromosomes. You could even say 23 types of chromosomes, right? Because chromosome 1 is not the same as chromosome 2. They have different genes on them. All right. All right, so when sperm and egg unite and the sperm fertilizes the egg, we get a diploid cell with 46 chromosomes. That's two of each chromosome, or 23 pairs of chromosomes. So if we go back to the karyotype, just to point this out, again, 23 pairs. A pair is 2, multiply 2 by 23, you get 46. So your somatic cells have 46 chromosomes arranged in 23 pairs, okay? The sex cells, the sperm and the egg, they only have 23 chromosomes. They don't have 23 pairs. They have 23 singular chromosomes, one of each chromosome, okay? One copy of chromosome 1, one copy of chromosome 2, one copy of chromosome 3, on down, one copy of either the X or the Y chromosome, but they don't have both X and Y. All right, so those are the sex cells. They have 23 individual chromosomes. All right. So the egg has 23, the sperm has 23, and when they fuse together, the resulting zygote is going to have 46 chromosomes. Okay? And so meiosis is important for this reason. We need to cut down the number of chromosomes in our sex cells by half so that when our sex cell fuses with someone else's sex cell, the resulting zygote is going to have the right number of chromosomes, 46. Now, my skin cell has 46 chromosomes, 23 pairs. Okay, my skin cell has 46 chromosomes. If my sperm cell also had 46 chromosomes, and my partner's egg cell had also 46 chromosomes, the resulting zygote would have 92 chromosomes. We would be doubling the number of chromosomes every time we reproduce. And that's not only untenable, it's dangerous, of course, because we mentioned gene dosing issues. You need to have the right number of chromosomes. All right, some gene dosing issues are fatal, some are not. As we can see, obviously, with trisomy 21, Down syndrome is not fatal. All right, so meiosis is similar to mitosis, but there are two divisions, meiosis 1 and then meiosis 2. Okay. All right, so we've talked about the chromatids and the centromere. Somatic cells are diploid. That is my skin cell, my brain cell, my muscle cell. They're all diploid, meaning they have nuclei that contain two sets of chromosomes. That is 23 pairs of chromosomes. Meiosis 1 is going to separate the homologous chromosomes. Okay. And we're also going to see some crossover where genes from one of your chromosome 1s may cross over onto the other chromosome 1. Okay, and we'll look at that. The resulting cell is going to be haploid. It'll have 23 pairs of chromosomes, but those chromosomes will be identical to one another. Okay. And then in meiosis 2, the cells are still haploid, and when they divide, the resulting cells will also be haploid. And we'll kind of take a look at how that happens. Haploid means you have one set of chromosomes as opposed to two. Diploid means you have two sets of chromosomes. All right, so here is a germline cell, one of the stem cells that divide and produce either egg or sperm cells. And it has 46 chromosomes arranged in two pairs. Okay. We go through meiosis 1. Okay. We're going to divide, and then the daughter cells are technically haploid. Technically haploid. What that means is that they're going to have one chromosome 1, and then they're going to have another identical chromosome 1. Okay, so even though there are two chromosome 1s here, because the two are identical, they only have one version of chromosome 1. Even though there are two chromosomes, they only have one version of it. I hope that makes sense, because this is some pretty heady stuff. All right, so we go through our first meiotic division, and we wind up with, yes, haploid daughter cells that are going to have two identical copies of each chromosome. Now here we're going to go through some crossover. We'll take a look at that in a little bit. Then the cells divide again. We go through meiosis 2, and this time we're not replicating the DNA. We're just dividing these daughter cells into two more daughter cells. Okay, we're not going to replicate the DNA. We're just dividing it into two more daughter cells. All right, same thing down here. And so the end result, ideally, well, I say ideally, on paper, the end result should be 4 sex cells, 4 sperm or 4 eggs. The truth of the matter is that this is exactly how it works for sperm. Eggs are a different story. With eggs, only one of these cells will develop into an egg. The other 3 will develop into shrunken little cells called polar bodies, and those polar bodies will be destroyed. All right, so the stages of meiosis 1 are the same as mitosis, at least name-wise. We've got prophase, metaphase, anaphase, telophase, and we signify it's meiosis 1 with prophase 1, metaphase 1, anaphase 1, telophase 1. All right. So synapsis is the pairing of the homologous chromosomes. That's where we line them up next to each other down the middle. And then here we have crossover during prophase 1. Crossover kind of looks like this. And so here you've got two sets of sister chromatids. This is 2 copies, we could say, of chromosome 1, 2 copies of chromosome 2. And then they're reaching over, their arms are reaching over, so that in the end we have, here's a normal chromosome 1, or a normal blue chromosome 1. And then here's a chromosome 1 with a little bit of the red chromosome 1. Over here, here's a red chromosome 1 and another red chromosome 1 with a little bit of the blue. And so these two regions are identical except in the gene versions that are on this chromosome. And so we used the hair color example earlier. It's a made up example, but let's go ahead and use it here because it's really simple. Okay. So we could say on let's say this is chromosome 1, and let's say chromosome 1 right here has the gene for hair color, And let's say the blonde one is on blue, and let's say red hair is on red. Okay. All right. Now, over here, there's a little bit of crossover. Okay. And what we wind up with is a chromosome here that has all of the blue chromosomes except for this region, which now contains the gene for red hair. And over here we have a red chromosome with all the normal red chromosome versions of genes, except for this region, which contains the gene for blonde hair. All right. I'm amazed there are no questions yet because this is kind of confusing stuff. All right, so to continue on, metaphase again, after crossover, we're going to line them up down the middle, and then we're going to pull the homologous chromosome pairs apart. And so what that means is that where before, let's go ahead, you've got one version of chromosome 1 and you've got another version of chromosome 1 normally. We don't normally use this. This isn't an official term in the sciences, but just to simplify it, why don't we call them chromosome 1A and chromosome 1B? Let's just do that. And so let's say chromosome 1A, to go back to this picture, let's say 2 copies of chromosome 1A is going to wind up in this daughter cell, and 2 copies of chromosome 1B will wind up in this daughter cell. Okay? We don't normally use that terminology 1A, 1B, but it actually would kind of simplify this idea a little bit to call it the A version and the B version. The A version, let's say, goes up here, and then the B version goes down here. Okay, so you got 2 copies of the A version up here. All right, and so both of the daughter cells will get a copy of the A version. You got the 2 copies of the B version down here, and so after the second mitosis, both daughter cells will get a copy of the B version. Here's another example of crossover. This cartoon version is a little prettier than the one I made on the last slide. You can see the arms, they cross over, and they're identical in structure. They're identical in which genes are on this leg of the chromosome. What's different are the versions of the genes. So after meiosis 1, we move into meiosis 2. And so in meiosis 2, that's where we're going to see prophase 2, metaphase 2, anaphase 2, telophase 2. And meiosis 2 is almost identical to mitosis. So here the sister chromatids are going to line up down the middle. In metaphase, they'll condense in prophase. Metaphase, they'll line up down the middle. Anaphase, they'll be pulled apart. Telophase, we see the nucleus reform. That's followed by cytokinesis, where we get 2 daughter cells. All right. And so again, on paper, what we should wind up with is 4 daughter cells. Now, all 4 daughter cells are haploid. Okay, all 4 daughter cells are haploid, meaning they have 1 set of chromosomes. Okay. Why is it important that they be haploid? Because you need to have half the normal number when this cell meets another sex cell. Oh, excuse me. Right. So when sperm meet egg, sperm and egg both need to have half the number of chromosomes so that when they unite, the resulting zygote has 2 sets, the normal number of chromosomes. Okay. All right. Good. All right, so this is a lecture on reproduction, and that's not really what we're getting into tonight, just mitosis and meiosis. So I'm going to skip through a lot of this. Here you can see meiosis for sperm cells looks very much like the diagram we just saw. Go through the first meiotic division, we form the spermatocytes, the secondary spermatocyte. Now the secondary spermatocyte again, let's say this is chromosome 1 and chromosome 2. It's got 2 identical copies of chromosome 1, 2 identical copies of chromosome 2, and they're going to be different from the copies of chromosome 1 in this cell and chromosome 2 in this cell. Now they go into meiosis 2, which is very similar to mitosis, where they just line up down the middle normally. Anaphase, they are pulled apart. Telophase, we get the new nucleus. And in the end, we get 4 haploid sperm cells, okay, with half the normal number of chromosomes. These secondary spermatocytes, which are made after meiosis 1, are technically haploid because even though they have 2 copies of chromosome 1, the 2 copies are, again, identical. Okay, so they only have one version of chromosome 1. All right, and moving on, we're just going to keep going down to the female reproductive. Female reproductive is a little, well, oogenesis is a little more complicated. So here we start with what's called a primary oocyte. It goes through meiosis 1, okay? And just like we saw in the sperm cell, we're going to get up here 2 copies of chromosome 1 that are identical, 2 copies of chromosome 2 that are identical. This cell is haploid. Down here, same thing, 2 copies of chromosome 1 and 2 copies of chromosome 2 that are identical. Okay, the 2 chromosome 2 copies are identical to each other. The 2 chromosome 1 copies are identical to each other. So this cell is also haploid. Now, you may notice a difference in size, right? The secondary oocyte and the polar body are different in size. The secondary oocyte, during cytokinesis, when the cell actually divided, the secondary oocyte received the majority of the cytoplasm and the majority of the organelles. The polar body is left with very little down here. And so you may see a second meiotic division, but probably not. And if you do see a meiotic division, you'll see a first and a second polar body, but over time, the polar bodies will disintegrate. They'll break apart. So it doesn't really matter if you have a second meiosis or not for the polar body. This just explains where the first and second polar body go. They break down. Now up here, we've got the secondary oocyte, and the secondary oocyte is sitting around waiting for a sperm to arrive. When the sperm arrives, penetrates the egg cell and fertilizes it, okay, the sperm cell is going to lose its tail, and the head up here, which is basically a nucleus, is going to enter the secondary oocyte. And here's just something really interesting. The egg does not go through meiosis 2 until the sperm cell shows up. When the sperm cell shows up, then it goes through meiosis 2. Okay? Anyway, it goes through meiosis 2, and, one set of chromosomes is going to remain with the sperm cell, and the other set of chromosomes gets kicked out in another polar body. And this polar body is also going to break down. And what we're left with in the end is the zygote. Now, the zygote has 46 chromosomes. 23 of them came from the sperm cell, and 23 of them came from the secondary oocyte. Okay? And what we're left with, again, yes, a zygote with 46 chromosomes, 23 pairs of homologous chromosomes. Okay? All right, here you can see an electron micrograph showing the egg cell and then a little polar body over here. This is before division. The polar body is gonna be released and it'll break down. All right. Good, good. So that is meiosis. Do we have any questions so far? Please throw me something. I know, mitosis and meiosis are not fun. They are pretty complicated ideas, but they're important. They're important. All right, so let's talk about trisomy 21 just for a second. I said we'd come back to it when we talked about meiosis, because it's technically a meiotic thing. All right, so here, let's look at this diagram. This could be sperm or egg, because this is an idealized graph. Anyway, uh, so in anaphase, which is where, no, I'm sorry, in metaphase, which is where the chromosomes line up, okay, these spindles have pulled them into position, okay, then in anaphase, it's going to pull them apart to opposite ends of the cell. Let's go ahead and say it occurs during meiosis 2, okay, and let's say this is chromosome 21 right here. Okay, you have a copy of chromosome 21 that's supposed to go up to this side for this daughter cell, and you got a copy of chromosome 21 that's supposed to come down to this side for this daughter cell. Okay. Now, occasionally there are errors in the division of the chromosomes, and so occasionally, this doesn't happen often. In fact, most of the time it is fatal. Trisomy 21 is interesting, Down syndrome is interesting because it's one of the rare examples where this sort of thing is not fatal. Anyway, so during this division, during anaphase where they're pulled apart, okay, something goes wrong, and then one of the daughter cells gets 2 copies of chromosome 21, and the other daughter cell gets no copy of chromosome 21. And so this cell, if it ever fertilizes or gets fertilized, assuming it has no copy of chromosome 21, it will spontaneously degenerate. It will essentially die and break down. The other one, which got 2 copies of chromosome 21, let's say this is a sperm cell, if it goes and fuses with a normal egg cell, which has 1 copy of chromosome 21, the 2 from the sperm and the 1 from the egg unite to form a zygote that has 3 copies of chromosome 21. And again, chromosome 21 is a gene dosing condition. You have too many copies of chromosome 21, and so you're going to have too many copies of whatever proteins are made by the 21st chromosome. All right, and so that affects development. And it all goes back to an error in the division of chromosomes during anaphase. All right, so I finished talking about Down syndrome. We've talked about just about everything I wanted to. Good. So let's finish by talking about the assignments. Okay. In the learning content. Okay. For week 5, I apologize. I posted that a little bit late. You can see we've got a video. We've got a Learn Smart as usual. And then we have the mitosis activity, which is some answering questions, some labeling, that kind of thing. We move on to learning content for week 6. Meiosis, you're going to see a very similar thing. We've got a Learn Smart, we've got a video, and then another meiosis activity. For weeks 5 and 6, the meiosis activity is replacing a discussion board. Okay, so don't worry about the discussion board for last week or this week. They're being replaced by these mitosis and meiosis activities. Okay. All right, so that's it for me. I appreciate you guys tuning in. Appreciate you guys watching these videos. And if you have any questions, don't hesitate to send me an email. I love answering content questions. I love helping people understand this better. I know I don't always do the best job of explaining it the first time. So if that's the case and I've confused you in any way, I apologize, and send me an email. I will clear it up. I will spend all day, if I have to, organizing my thoughts in a logical manner so that I can better explain this to you. Okay, you get the picture. All right, thanks for tuning in, and we'll see you guys next week.